Okay, so now you have an understanding of the experiment that was performed for the data science challenge. And now we're going to move into the last section, which is what does that data look like and how do we work with it? So you're going to be able to load the data into the Python environment using these packages scanpy and Anndata. And so scanpy is a Python package that's commonly used in the single cell analysis community, and it's very streamlined and hopefully easy to use for you. And within scanpy, there's another package called Anndata, which allows us to load in the data and organize it in a very sensible way. So scanpy and Anndata are going to be the main tools that you can use to at least get the data into your hands and start to work with it. And so I'll give you a few lines of code that might help you along the way. So, when we are going to load the data, we're going to import scanpy. And when you import scanpy, that will bring along Anndata. And then other useful packages could be NumPy or pandas. Okay, so Anndata is short for annotated data, and it's a way to structure the data. And the two pieces of Anndata that we're going to care about the most are what are called X and obs or observation. So X is going to be where the gene expression matrix is stored. So if your Anndata object that you load up is called Adata, then the gene expression matrix will be loaded by just typing adata.x. And similarly, for every single cell in the gene expression matrix, we're also going to have metadata. And that's going to be stored in the obs or observation data frame, which is a pandas data frame. And obs will have for every single cell what perturbation that cell received, as well as what cell state we assigned that T cell to. So we've already gone through and analyzed this data and assigned T cell states to every single cell in the data that we're going to give you. Now, to load up the data, we're going to give you an object that's an H5AD file, and you can load it up using the read H5AD function, and that will give you your Anndata object, which we're just going to call Adata in these slides. In that object, you can just look, and it has a gene expression matrix in it, as well as an observation data frame. So let's look at these individual pieces separately. So the gene expression matrix is stored in adata.x, and this is a sparse matrix. And it's a sparse matrix because, to remind you, single-cell RNA sequencing data, the gene expression matrices have a lot of zeros. So you can actually store the matrix in sparse format. And the matrix has dimensions observations by variables. So the observations are the number of cells that were profiled, 31,000 here. And the number of variables or features is the number of genes that were measured, 15,000. Now, this number, 30,000 cells, is different from the number I told you earlier, and that's because we've done quality control to the data, and we'll talk a little bit about the steps that were done in the quality control, but this is the final number of cells you're going to be given. Now, to look at some of the values in that expression matrix, we can just convert to an array, and now you can see the expression values, and again, you can see lots of these zeros, which is why we store it in sparse format. Finally, we can load up the names of the cells and the names of the genes. So we have obs names and var names. So obs names, you see these very long strings that are describing the cells, and you'll see the string of A, T, Cs, and Gs, and that comes from the DNA barcode that was introduced to label the different transcripts from every single cell, and this goes back to lecture 2 where we introduced the single cell isolation techniques using microfluidics. So the names are not super important, but just so you're not wondering what these A, T, Cs, and Gs are. And then we also have the gene names, which are very interpretable. And within these set of gene names, there will be genes that we've introduced in the lecture, like TCF7 or PDCD1. So that's the Anndata.x matrix. And then we also have the Adata.obs, which is this pandas data frame, which has for each cell different metadata describing that cell. And the two main columns that we care about are the genetic perturbation the cell received. So for instance, this cell received a non-targeting perturbation, meaning that's a perturbation that doesn't affect the cell, shouldn't affect the T cell. And then this cell received a perturbation to TOX2 and TPT1 and so forth. And then we also have the T cell state that we assigned after doing analysis of this data. And remember, there are 4 functional T cell states in this challenge: the progenitor T cells, the effector T cells, the exhausted T cells, and the cycling T cells. And you'll find all of those in this metadata. Okay, so one of the things that we're going to be really focused on is for each genetic perturbation, what are the proportions of T cell states we find for that perturbation? So here's some code to simply calculate for every single genetic perturbation, what proportion of cells are in each T cell state. So for instance, this is perturbation 1, and we find that 28% of the cells for perturbation 1 are in the terminally exhausted state, and 26% of cells are in the progenitor state. And then we can look at the last perturbation, perturbation 63, and we find that 50% of the cells are in the progenitor T cell state and 28% are in the cycling T cell state. So this is just a simple code to calculate the proportions of T cell states for each genetic perturbation. Now I'll just show you some of the raw data. So here what we're looking at are all the genetic perturbations that were applied in Mark's experiment. And the y-axis is the number of cells in which that genetic perturbation was detected. And the perturbations are ordered by number of cells in which they were detected. And this is a log scale. And what you can see is that most cells have more than 100, most perturbations are detected in more than 100 cells. And we want the perturbation to be detected in multiple cells because increased sample size for a perturbation is going to give us a better sense of the effect of that perturbation. Single-cell RNA sequencing data is very noisy, so the more measurements we can make of the same perturbation across multiple cells, the more confident we'll be in inferring that perturbation's effect on gene expression. So fortunately, most perturbations are detected in many, many cells, but this is something to keep in mind, that the number of cells we look at kind of affects like how confident we are for inferring any perturbation's effect. So next, I just want to tell you what quality control and normalization was done on this data. So I want to remind you that when we introduce these perturbations into the T cells, I describe this as each T cell gets one genetic perturbation. Now, that's a little bit of a simplification. So ideally, we would like a T cell to just get one perturbation and not 0 perturbations or many perturbations. But actually, the number of perturbations that get introduced into a cell when doing this process of introducing perturbations using CRISPR-Cas9, the number of perturbations that get introduced into a cell follows a Poisson distribution, meaning that most cells only received one perturbation, but some fraction of the cells received 2 or more perturbations, and some fraction of the cells received no perturbations. So we do quality control to remove cells that received 2 or more perturbations because we are not powered to infer the effects of combinations of perturbations in this data that would take looking at hundreds of millions of cells. So we remove these cells here. We also remove cells that received no genetic perturbation. And you might ask, why do we do that? Well, part of the reason is that cells that receive no genetic perturbation, they might have not received a perturbation, but there might also be a technical reason that we weren't able to detect the perturbation. And we would like to model what unperturbed cells look like, but the experiment is designed such that we can use the non-targeting controls to model that distribution. So we don't really need these cells. We can actually just use the non-targeting control perturbations to infer what the T cell state distributions look like in an unperturbed state. And then finally, we remove cells where perturbations have very low counts because those are technically probably low quality cells. So that's the quality control. And then what we end up doing is for every cell, we normalize the total gene expression. So now the total gene expression is just going to sum to 1. And then what we do is we multiply all of that by 10,000. So now this total expression sums to 10,000. We add 1 and take the logarithm. OK, so once we've done this cell quality and normalization, we carry out the rest of the single-cell RNA sequencing analysis, where we do feature selection, dimensionality reduction using principal components analysis, and then graph-based clustering. OK, so here we have the results from the experiment that Mark performed where he did Perturb-seq in T cells. And so here we're showing a UMAP embedding of the 30,000 T cells that pass quality control. And to remind you, the UMAP embedding is calculated from the cell's principal component scores. And the idea is that it's preserving the similarity between cells that exists in high dimension, and it's projecting that down into 2 dimensions. So 2 cells are similar to one another if they have similar gene expression profiles in high dimension. And then if they are similar, we want them to be close to one another in 2 dimensions. So here, these 2 cells are similar to one another and have similar gene expression profiles. And then here, these 2 cells are close to one another, have similar gene expression profiles. So we calculate the UMAP embedding for all the 30,000 T cells that pass the final quality control. And then we color them by their cluster identity that we derived in the previous slides. So starting with progenitor cells, we can ask what genes are more highly expressed in this cluster relative to all the other clusters. One gene that will come up is TCF7, which we know marks the progenitor T cell state. We can also look at the terminal exhausted cells, and one gene that is more highly expressed in those relative to all the other clusters is PDCD1, and that helps us mark the terminal exhausted state. So we can do this for all the clusters, and this allows us to identify the 4 main T cell states we see in cancer, namely the progenitor state, the effector state, the terminal exhausted state, and the cycling state. Okay, so now we get to the really fun part of the data, which is we can link the gene expression we see in single cells in the cell state to the perturbation that that cell received. And we can ask, how do the perturbations affect the proportions of T cell states we see? So first, let's just calculate the overall T cell state proportions in all the data. So we see that there are 10% of T cells are in the progenitor state, 30% are in the effector state, 20% are in the terminally exhausted state, 30% are in the cycling state, and 10% are what we call other. Now let's look at what happens when we do a genetic perturbation. And I'm going to leave these perturbations unlabeled, but we're going to perturb a single gene and ask, for all the cells that receive that genetic perturbation, what's the resulting proportion of T cell states? And what we see is that the progenitor T cell state proportion went up, it's 40% now, and the terminally exhausted T cell state decreased. And this could be a really beneficial thing for cancer immunotherapy because, as we discussed before, we think progenitor states and effector states are more effective when responding to cancer. Now, in stark contrast to this, we can look at a different perturbation, perturbation 2. And here we're just showing all the cells that receive perturbation 2. And what you see is there's this big drop in the progenitor T cell state and a big increase in the terminally exhausted T cell state here. And so this you could think would be really bad in cancer because these terminally exhausted T cells are unable to really respond and control cancer. And then finally, we have these really clever negative controls where these cells all received a non-targeting control perturbation. And we can look at the proportion of T cell states there, and we can compare that to the genetic perturbations that actually affect a specific gene. And so we can always just do these comparisons between the negative control and the actual genetic perturbation to infer how a genetic perturbation affected the overall T cell state proportions. Okay, so now hopefully you're starting to see how all these challenges fit together. So the first challenge task is to predict the T cell state distribution. So remember, Mark measured 70 genes in his experiment. He made 70 genetic perturbations. We're only giving you 63 of those perturbations in the training set, and we're going to ask you to predict the effect of the 7 other genetic perturbations that are in the test set. And so these perturbations might increase or decrease the different states. So one perturbation, as we saw here, increased the progenitor T cell state, and another perturbation that we saw here increased the terminally exhausted T cell state. So we'll ask you to predict the effects of these perturbations on the T cell state proportions. Then in the second challenge, we're going to turn this problem on its head a bit, and we're going to ask, given that we want to maximize some T cell killing objective, what are genetic perturbations we could make to get there? So let's define both sides of that. So first, what do I mean when I say maximize T cell killing objectives? So one thing that we think would improve immunotherapy is if we had a higher proportion of T cell states in the progenitor state and the effector state, and a lower proportion of T cell states in the terminally exhausted state. So that's an objective that we want to maximize. And we think that's going to make T cells better at killing tumor cells. We think that's going to make CAR T cells less exhausted. And we think that's going to have benefit to checkpoint blockade therapies. So that's the objective function. And what we're going to ask you to do is think about all 20,000 genes that are out there and choose the best 3 genes that are going to maximize that objective. So what are the 3 genetic perturbations that could really give you more T cell killing? So one perturbation that seems like it's going in the right direction is maybe this perturbation 1, where we have more progenitor T cells. And a perturbation that seems like it's going in the wrong direction is this perturbation where we have more terminal exhausted T cells. And then finally, the last challenge will be to propose a quantitative metric to rank the efficacy of the different proposed perturbations. And so now we've reached the end of our introductory lectures. And we've gone through the biology behind this data science challenge, the technology that's enabling us to do these experiments, the experiment itself that Mark did to do Perturb-seq in melanoma, and how you can work with the data yourself and really get into the challenge. And so we're really excited about this, and we hope you are too. We can't think of anything more important to work on than making cancer immunotherapy better. So we thank you for your participation and hard work in this, and I just want to wish you good luck and have fun with this. Thanks.